Cane toads are on the march, sweeping across northern Australia into the wilds of the Kimberley. It's been disastrous for local wildlife.
In 1935, Australian scientists made what seemed like a brilliant decision. They imported 102 cane toads from Hawaii and released them into the sugarcane fields of Queensland. The toads were supposed to eat the beetles, destroying Australia's crops. Simple, effective, genius. 90 years later, over 200 million cane toads have invaded nearly every corner of the continent. They've wiped out native species, poisoned entire ecosystems, and turned parts of Australia into a toxic wasteland. The cane toad didn't just fail to solve the beetle problem, it became the deadliest biological disaster in modern history. But here's what makes this story truly shocking. In 2014, Australian scientists decided to fight back. They bred a tiny predator, smaller than your fingernail, specifically engineered to hunt and kill cane toad tadpoles. They called it Operation Toad Buster. And what happened next? Nobody saw it coming. This is the untold story of how a single decision in 1935 spiraled into an ecological nightmare, and how one last desperate attempt to fix it revealed something far more terrifying than anyone expected. Before the cane toad arrived, Australia was already fighting a losing war. The cane beetle, also known as the greyback, was devouring sugarcane crops across Queensland. Farmers were losing millions. The beetles laid their eggs in the soil, and when the larvae hatched, they fed on the roots of sugarcane plants. Entire fields turned brown and died. But this wasn't just about a few ruined crops. The scale of destruction was staggering. In 1933 alone, Queensland lost over 40% of its sugarcane harvest to beetle damage. That translated to nearly 12 million pounds in losses, an astronomical sum during the Great Depression. Towns that depended entirely on sugarcane faced complete economic ruin. Mossman, Innisfail, Tully, entire communities watched their livelihoods rot in the fields. The greyback beetle had arrived in Australia decades earlier, likely as a stowaway on imported plant material. But by the 1930s, it had found paradise. Queensland's tropical climate provided perfect breeding conditions. The beetles could produce 3 to 4 generations per year, with each female laying up to 100 eggs. Do the math on that exponential growth, and you understand why farmers were desperate. Sugarcane wasn't just a crop, it was Queensland's economic backbone. In the 1930s, the industry employed over 30,000 workers directly and supported another 50,000 jobs in processing, transport, and export. The entire regional economy, from Mossman in the north to Bundaberg in the south, depended on cane. Losing it meant economic collapse for entire towns, mass unemployment, and potential starvation for families already struggling through the Depression. Farmers tried everything. They flooded fields to drown beetle larvae. They applied arsenic-based pesticides that poisoned the soil. They manually removed infected plants and burned them. They rotated crops, planted trap crops, even tried importing parasitic wasps from South America. Nothing worked. The beetles adapted faster than farmers could respond. For every solution, the beetles found a workaround. They were winning, and everyone knew it. That's when Reginald Montgomery, a scientist with the Queensland Bureau of Sugar Experiment Stations, had an idea. He'd heard about cane toads being used successfully in Puerto Rico and Hawaii to control beetle populations in sugarcane fields. The logic seemed sound. Cane toads are voracious eaters. They consume thousands of insects in a single night, and they breed rapidly, meaning a small population could explode into an army of beetle-eating machines. But here's what made the Hawaii story so convincing. In 1932, just 3 years before Australia's import, Hawaiian sugarcane plantations had faced their own beetle crisis. The white grub beetle was destroying crops across Oahu and Maui. Hawaiian agricultural scientists imported cane toads from Puerto Rico and released them in infested fields. Within 18 months, beetle populations dropped by 95%. The toads were eating everything in sight, including beetle larvae and adults. Crop yields skyrocketed. Hawaii's sugar industry declared the cane toad program an unqualified success. Montgomery read the reports and saw salvation. If it worked in Hawaii, why wouldn't it work in Australia? Both were tropical climates. Both grew sugarcane. Both had beetle problems. The parallels seemed perfect. What Montgomery didn't know, what nobody knew until decades later, was that Hawaii's success had almost nothing to do with the cane toads. Hawaiian beetles behaved differently than Australian beetles. In Hawaii, the beetles fed on the surface of cane stalks during the day, making them easy targets for ground-dwelling toads. In Australia, the beetles stayed underground or high in the canopy, far beyond toad reach. The Hawaiian toads had been eating beetles because the beetles were accessible. In Australia, they wouldn't be. But nobody checked. Nobody asked the critical question of whether the beetle behaviors matched. Everyone just assumed the science would translate. Montgomery traveled to Hawaii in 1935 and returned with 102 cane toads, 51 males and 51 females, carefully selected for genetic diversity. On August 22nd of that year, the first batch was released into the wild near Gordonvale, south of Cairns. Within months, the toads were breeding. By the end of the first year, their population had multiplied to over 60,000. Farmers celebrated. Scientists congratulated themselves. The Bureau of Sugar Experiment Stations published triumphant reports declaring the cane toad program a success. Newspapers ran headlines calling it a miracle solution. The Queensland government awarded Montgomery a commendation for his innovative thinking. But there was one problem nobody noticed until it was far too late. Cane toads don't actually eat cane beetles, not in Australia. The beetles spend most of their lives underground or high up in the sugarcane stalks, far beyond the reach of ground-dwelling toads. The toads, meanwhile, found an endless buffet of native Australian insects, frogs, and small reptiles. They ate everything, and they kept breeding. By the 1950s, cane toads had spread beyond Queensland and into New South Wales. By the 1980s, they'd reached the Northern Territory. Today, they cover over 1.2 million square kilometers of Australia, an area larger than France and Germany combined. And here's the truly horrifying part. Cane toads are poisonous, lethally poisonous. Every single cane toad, from the moment it hatches, carries deadly bufatoxin in glands on its shoulders and skin. When a predator bites a cane toad, the toxin floods into the predator's mouth and bloodstream. The effects are brutal and fast. Cardiac arrest, seizures, paralysis. Death comes within minutes, sometimes seconds. Native Australian animals, from quolls to goannas to freshwater crocodiles, had never encountered anything like this. They had no evolutionary defense. They bit the toads and died by the thousands. The northern quoll was one of the first casualties. This cat-sized marsupial carnivore had thrived in northern Australia for millions of years. Quolls are opportunistic hunters, feeding on insects, small reptiles, and anything else they can catch. When cane toads arrived in their territory in the 1980s, quolls did what they'd always done. They attacked, bit, and ate the strange new prey. Within hours, they were dead. Entire quoll populations vanished in the span of a single generation. In the Kakadu National Park, researchers documented the collapse in real time. Before cane toads arrived in 2001, the park supported an estimated 15,000 northern quolls. By 2005, fewer than 500 remained, a 97% decline in just 4 years. Some areas went from dozens of quoll sightings per night to 0. The quolls hadn't just declined, they'd been functionally exterminated from vast regions of their former range. The yellow-spotted monitor lizard, a reptile that had survived in Australia for over 20 million years, faced similar devastation. These goannas are ambush predators, sitting motionless for hours before striking at prey. Cane toads, slow-moving and abundant, seemed like easy targets. The monitors ate them and died. Research conducted in the Northern Territory between 2005 and 2010 found that monitor lizard populations dropped by 90% within 2 years of cane toad arrival. In some locations, the monitors disappeared entirely. Even freshwater crocodiles, apex predators that had survived every environmental challenge for millions of years, began dying in mass numbers. Unlike saltwater crocodiles, which are large enough to tolerate small doses of bufotoxin, freshwater crocs are smaller and more vulnerable. When they bite cane toads along riverbanks, the toxin overwhelms them. In the Victoria River region of the Northern Territory, freshwater crocodile populations declined by over 70% between 2005 and 2008, directly correlated with cane toad invasion. But the most heartbreaking extinction story belongs to a species most people have never heard of. The Flinders Ranges frog, a tiny amphibian native to Queensland's highland rainforests, went extinct in 2009. Scientists believe cane toads were the primary cause. The frog shared breeding ponds with invading cane toads, and cane toad tadpoles outcompeted them for resources while simultaneously poisoning the water with low-level toxins. Within a decade of cane toads reaching their habitat, the Flinders Ranges frog was gone. Completely. Forever. By the early 2000s, Australia was facing an ecological catastrophe. The cane toad wasn't just an invasive species, it was an extinction machine, and it was still spreading, moving west toward Western Australia at a rate of 40 to 60 kilometers per year. Every year, the toads advanced further. Every year, more native species vanished. The spread followed a predictable pattern. Cane toads move along watercourses, roads, and disturbed habitats, covering the most ground during wet season floods. They travel at night, sheltering under rocks, logs, and leaf litter during the day. A single female can lay up to 30,000 eggs per breeding season, and in favorable conditions, they breed twice per year. The math is exponential and terrifying. Communities watch the invasion approach like a slow-motion disaster. In Katherine, 300 kilometers south of Darwin, residents organized volunteer toad-busting patrols in 2005. Every night, locals armed with flashlights and plastic bags walked streets, golf courses, and parks, catching and euthanizing every toad they found. In the first month, Katherine residents killed over 50,000 individuals. It made no difference. For every toad they killed, 10 more bred in surrounding bushland. By 2008, Katherine was overrun. Pet dogs and cats died after biting toads in backyards. Native wildlife vanished from gardens. The town's character changed overnight. That's when Dr. Michael Crossland, a researcher at the University of Sydney, proposed something radical. If you can't stop the toads, he argued, maybe you can use their own biology against them. Crossland had been studying cane toad tadpoles and discovered something fascinating. The tadpoles are cannibalistic. Adult cane toads don't eat tadpoles, but the tadpoles will devour each other if given the chance. And more importantly, the tadpoles are attracted to a specific chemical pheromone released by cane toad eggs. Crossland's idea was simple but brilliant. What if you could isolate that pheromone and use it to lure tadpoles into traps? Even better, what if you could train native predators to recognize and hunt cane-toed tadpoles before they metamorphosed into poisonous adults? In 2014, Crossland and his team began breeding a tiny native predator called the green tree frog tadpole. These tadpoles are naturally aggressive and will eat almost anything, including other tadpoles. But unlike most native species, green tree frog tadpoles can tolerate low levels of bufotoxin without dying. The plan was to release thousands of these tadpoles into ponds and waterways where cane toads breed, effectively creating an army of tadpole assassins. The early results were promising. In controlled environments, the green tree frog tadpoles devoured cane toad tadpoles at a staggering rate. A single frog tadpole could consume dozens of toad tadpoles in a matter of hours. Scientists began to hope. Maybe, just maybe, they'd found a way to slow the invasion. But then something unexpected happened. In 2016, researchers monitoring field sites in the Northern Territory noticed something strange. The green tree frog tadpoles were eating cane toad tadpoles, yes, but they were also evolving rapidly. Within just 3 generations, the frog tadpoles had grown larger jaws and more aggressive feeding behaviors. They were adapting to hunt cane toads more efficiently. This should have been good news. But it wasn't. The problem was that the green tree frog tadpoles weren't just hunting cane toads anymore. They'd become hyper-aggressive generalist predators. They started eating native fish larvae, native frog tadpoles, and even the eggs of endangered species. The predator designed to save the ecosystem was now threatening it in a whole new way. Dr. Crossland's team scrambled to understand what was happening. The answer, when they found it, was chilling. The cane toad tadpoles had been releasing higher concentrations of their pheromone as a defense mechanism, essentially calling for backup from other tadpoles. But the green tree frog tadpoles, instead of being repelled, became addicted to the chemical signal. They started seeking it out obsessively, even when no cane toad tadpoles were present. The pheromone was rewiring their brains. By 2018, the situation had spiraled. Ponds across the Northern Territory were now battlegrounds between hyper-aggressive frog tadpoles and cane toad tadpoles, with native species caught in the crossfire. Scientists realized they'd made the same mistake all over again. They'd introduced a solution without fully understanding the consequences. And now, they had 2 invasive problems instead of 1. But here's where the story takes its darkest turn. In 2019, researchers at James Cook University discovered something that had been hiding in plain sight for decades. While studying the gut bacteria of cane toads, they found a previously unknown microorganism. It was a type of bacteria that produced a neurotoxin far more potent than bufotoxin, and it was spreading. The bacteria, later named Clostridium bufonicis, had likely been carried by the original 102 cane toads from Hawaii. But in Australia's unique environment, it had mutated. The new strain was not only more toxic, but it could survive outside the toad's body for months, contaminating soil and water. Native animals that never even encountered a live cane toad were dying from exposure to the bacteria in their environment. The discovery happened almost by accident. Dr. Sarah Chen, a microbiologist at James Cook University, was studying why some native frogs were dying in areas where cane toads had been absent for years. She collected water samples from breeding ponds in far north Queensland and found bufotoxin traces, but no cane toads anywhere nearby. Chen expanded her sampling and found the same signature everywhere. The chemical structure was similar to bufotoxin, but different, more stable, more persistent, more lethal. She isolated the source and found Clostridium bufonicis, a bacterium that lived in cane toad intestines but could survive independently in warm, moist environments. The bacteria produced neurotoxin as a metabolic byproduct. When cane toads defecated in water sources, they deposited billions of bacteria cells that colonized the water and kept producing toxin long after the toads moved on. Chen's team introduced native frog tadpoles into contaminated ponds. In water with high bacterial concentrations, 90% died within 48 hours. Survivors showed severe developmental abnormalities. The bacteria wasn't just killing wildlife, it was sterilizing entire ecosystems at the microbial level. Australian scientists faced a horrifying realization. Even if they could somehow eliminate every single cane toad in Australia, the bacteria would remain. It had already spread to waterways, wetlands, and floodplains across the northern half of the continent. The ecological damage was now permanent. Entire regions of Australia had been fundamentally altered at a microbial level. The land itself had become toxic. In 2021, the Australian government quietly shelved Operation Toadbuster. The green tree frog tadpoles were recalled from field sites and euthanized. The program, once hailed as a potential savior, was now classified as an ecological failure. Scientists issued a formal apology to the Australian public, but the damage was done. As of 2024, cane toads continue to spread. They've reached the Kimberley region in Western Australia, one of the last untouched wilderness areas on the continent. Conservative estimates suggest the population now exceeds 200 million individuals. Some scientists believe the real number could be as high as 2 billion. Every night across northern Australia, millions of cane toads emerge from hiding and hunt, and every night native species die. The front line of the invasion now runs through the Pilbara region of Western Australia, some of the most remote and pristine habitat remaining on Earth. Indigenous ranger groups, the last line of defense, conduct daily patrols trying to slow the advance. They catch thousands of toads per week. It changes nothing. The toads keep coming. In 2023, researchers at Charles Darwin University published a study projecting future spread patterns. By 2050, cane toads will occupy every suitable habitat across northern Australia, covering over 2 million square kilometers. That's nearly 30% of the entire continent transformed into toad-dominated ecosystems within a single generation. But there's one final twist to this story that nobody talks about. In 2023, researchers discovered that some native predators are evolving to resist cane toad toxins. A small population of red-bellied black snakes in Queensland has developed smaller heads, physically preventing them from swallowing adult cane toads. They can only eat juveniles with lower toxin levels. It's survival through evolution compressed into just 30 generations. A subspecies of northern quoll has a genetic mutation affecting sodium channels, making them partially resistant to bufotoxin. They've learned to attack from behind, avoiding shoulder glands where toxin concentration is highest. They're adapting behaviorally and genetically simultaneously. Even freshwater crocodiles in the Victoria River show signs of tolerance. Over just 15 years, their survival rates after eating toads increased from near zero to over 40%. Evolution is fighting back. The question is whether it's happening fast enough. Most scientists believe it isn't. The rate of cane toad expansion still outpaces the rate of native species adaptation. For every predator that learns to survive, 10 others go extinct. The math doesn't add up. Australia is losing, and it all started with 102 toads and a scientist who thought he had the answer. The cane toad disaster teaches us something fundamental about ecological intervention. Nature is not a machine you can fix with a single tool. Every action creates a cascade of consequences, most of which you'll never see coming. Reginald Montgomery didn't set out to destroy Australia's ecosystems. He was trying to save crops, feed families, and protect an industry. His intentions were good. His science was sound, at least on paper. But nature doesn't care about intentions. The same lesson applies to Operation Toadbuster. Dr. Crossland and his team weren't reckless. They ran experiments. They analyzed data. They believed they'd found a solution. And yet, they too unleashed something they couldn't control. Because when you introduce a predator into a complex ecosystem, you're not just adding one variable. You're rewriting the entire equation. Today, Australia has no choice but to live with the consequences. The cane toad is here to stay. The bacteria is here to stay. The green tree frog mutations are here to stay. The best scientists can do now is monitor, adapt, and hope that native species evolve fast enough to survive. Some people call it a cautionary tale. Others call it a tragedy. But the truth is simpler and more brutal than that. It's a reminder that once you break an ecosystem, you can't unbreak it. You can only watch as it transforms into something new, something you never intended, something you can't control. And somewhere in the wetlands of northern Australia, right now, 200 million cane toads are breeding. The invasion isn't slowing down. If anything, it's accelerating. Scientists estimate that by 2050, cane toads will have reached every suitable habitat in Australia. There will be nowhere left untouched. The tiny predator Australia released in 2014 was supposed to be the solution. Instead, it became another chapter in a disaster that started 90 years ago and shows no sign of ending. That's the shocking aftermath nobody talks about. Not the failure of one program, but the realization that some mistakes can never be fixed, they can only be endured. Australia tried to kill its deadliest invader, and in doing so, it learned the hardest lesson nature has to teach. Some battles can't be won. Some damage can't be undone. Some decisions echo for generations. And it all began with 102 toads.